Okay, welcome back. I wanted to go through part two of the immune system. I've had some issues with recording and Panopto, and so I do apologize for that. Hopefully, we've got it remedied. I ended up switching to YouTube, so you'll be able to access these through my YouTube channel now. So probably should have done that from the beginning, but they do encourage or have encouraged us to use Panopto. So anyhow, all right, adaptive immunity. Real briefly, part one discussed primarily innate immunity, these non-specific aspects, including epithelial barriers, phagocytic activity, inflammation, complement, and NK cells. We do see, and we will see today, the complement, or in this video, the complement can be innate or adaptive. So these aren't necessarily cut and dry. I suppose the lines of defense and is it adaptive? Is it innate? For the most part, epithelial barriers are going to be non-specific. We're born with them. They're part of the innate immunity. Phagocytes, similar manner. Inflammation, complement, NK cells. We're born with the ability to do these items. We're born with the chemistry to see these processes occur and these specific cells involved in immunity. So in that way, they're innate. However, there is some chemistry adaptation that can occur regarding age, exposure, environment, that type of thing. So anyhow, lines of defense, we looked at the first line of defense being primarily these epithelial barriers, whether they're mechanical or chemical. When we get into the second line of defense, we start to see phagocytic activity, some inflammatory features, adaptive aspects start to kick in, including complement chemistry, which we'll talk more about in this slideshow. But anyway, third line of defense then does start to get into some deeper B cell antibodies, effector T cells, more specific immunity. So COVID-19, obviously coronavirus, whatever you want to call it, has been in the forefront, obviously, for several months. We have a better idea, we think, of what it may be doing and maybe how to treat it and maybe even prevent it. So I'll mention a few things here and there in this video as well regarding COVID-19. And how some of the research and some of what we're going to look at with this immune system chapter can apply. So one of the items initially that applies is when we look at this innate immunity, the first line of defense has been breached with any infection. So COVID-19 is no different. It's a coronavirus. The corona part are these little spikes. They call them S proteins or spike proteins. They're the surface markers of the virus. So it's these spike proteins that are attaching to some of these epithelial barriers, triggering some sort of inflammatory response. The target that we're finding is a specific protein on the surface of really cells throughout the body. You've maybe heard of or seen some of this ACE2 or ACE2, what they're called angiotensin converting enzymes. And I'll put some links in at the end of this show that will take you to a couple of really good websites that break down some of what I'm discussing. We're going to talk about it in more detail in the next module when we do cardiovascular. We're going to look at urinary. We're going to look at acid-base, pH, fluid, electrolyte balance. So this ACE or ACE2 enzyme is something that we'll see later. The angiotensin, we're going to see again later. So I don't want to get into too much depth with it, but I do want to introduce you to, as we go, we'll look deeper into it, but I want to introduce you to some of the very basics of what we're seeing. And we see this with a lot of different viruses. Now, the flu virus, when it has a different appearance, but it does attack some of the similar markers that we see on the cell surfaces. Now, typically, we call those target cells, just like we saw with the endocrine system. So viruses have target cells. So, and even more specific target structures on cells. So even more specific, really, than just target cells. So, but anyway, the flu virus targets some of our mucus cells, some of these epithelial barriers. Usually that flu is fast acting, the flu virus, within a couple of hours even of that exposure. And really over the first couple of days, we do see most of the target cells destroyed. So that first line of defense starts to get broken down. But the phagocytes, also part of that first line of defense, and that innate, now we're into the second line of defense. The phagocytes of our innate immunity, and as the virus is destroying target cells, they'll kind of clean things up, and the flu virus is still maybe going to be around after a couple of days. However, the target cells have not been replenished. So usually after about 3 days, that third line of defense, which got triggered on day one. So third line of defense got triggered when the infection occurred. However, that third line of defense, which we think of as being that adaptive immunity, is really not prepared yet to send in the troops. So that usually takes 3 to 5 days. So anyhow, by day 3 of a flu infection, these guys are going to be ready to come in and really destroy and take care of through antibodies or effector T cells, really just to eliminate whatever virus is left. And that also is, as these guys come on the scene and start kind of cleaning things up, that will also trigger mitotic events and the regeneration of our first line of defense. So that progression and the timing works pretty well. That's why the flu virus, generally, they tell us we just have to wait it out. We need to rest. We need to hydrate. We need to just shut it down for 3 to 7 days. And after that, day 7, we're feeling, usually we should be feeling better. And again, it's because of the timing of our immune system. The innate immune system is attacked first. It kicks in first. It's short. The first exposure, within 12 hours, we've already started to take care of the infection. So we've allowed that second line of defense to start to come in. We see some inflammation. Again, usually that first 24 to 48 hours, there's the fever, the inflammation. That's a lot of second line of defense activity going on. We're kind of waiting for these adaptive immunity features in the third line to kick in. And again, that takes a little time. And again, in the meantime, the body has not started to regenerate more target cells. So the flu virus is still in there, but it doesn't have the targets. So the thing about coronavirus or COVID-19 is it comes in and it's a lot slower. It takes maybe 6 days to slowly kind of chip away and activate to get the third line of defense even, it's going to start going after 3 days. But the virus is still it's still attacking epithelial cells, or that first line of defense. The body has started after day 5 or 6 has started regenerating epithelial barriers, the first line of defense. In essence, target cells are being regenerated and the virus is still in the body or in the system. So whereas with the flu, we see the target cells, our own cells, the epithelial cells, they are they're not as, the timing is a lot faster with the flu. 1 or 2 days, it lingers, and the adaptive immune system has kicked in and eliminated it. And by day 3 or 4, our target cells have been remodeled. With coronavirus, day 3, day 4, we start remodeling our epithelial barriers, and we're adding more target cells, but the virus is still there. It hasn't been eliminated yet by the adaptive immune system. So they're researching whether we could slow the response time of the adaptive immune system even further. So allow the innate immune system, give it 6 days to give the coronavirus 6 to 7 days to work on the innate immune system and destroy the target cells, and then trigger the adaptive immune system to come in and destroy the lingering viruses that have no more target cells. If you eliminate the target cells and you still have the virus, that's where this adaptive immune system can basically come in and clean things up. Once it cleans everything up, the body then regenerates its cells, its first line of defense, and you don't have, there's at that point, there's no more virus. The virus has been eliminated by the adaptive immune system. And we have a memory of it as well. So if the target cells were to receive that virus, we would be ready quicker. And we'll see that later too, when it comes to second exposure of something, of a pathogen. So I don't know if any of that made sense. It probably didn't, but that's okay. It may by the end of this. I think the idea is I'll put a link to a scientific research article that goes through this process of describing how the flu virus works. Again, 1 to 2 days wipes all the target cells out, so that it still lingers, the virus, but after 3 days, we're ready to attack it. With COVID, 6 days it takes for the target cells to be eliminated, maybe even 7 days. Well, at day 3, we've already started sending our normal adaptive response. And so the adaptive immune system is destroying what it can. But in the meantime, there's still more target cells that are being attacked. So it's a slower moving virus. So that's what all that means. So anyway, I don't want to belabor it. I probably have belabored it enough already. So I'm kind of going to zoom through some of the adaptive stuff. I think these videos can get a little bit tedious for you, so I'll try to keep it brief and to the point. So adaptive immunity, again, part of that third line of defense. We're going to see B cell and T cells. We know this already. We've talked about it a little bit. We know that the B cells are going to be coming from directly from the bone marrow, maturing in the bone marrow and producing antibodies. So B cells produce the antibodies. T cells are born in the bone marrow and then they mature in the thymus gland and they produce T cells. We'll see a couple different types of T cells that are good. They're going to attack pathogens in a different manner. So let's take a quick peek at these strategies. We've got the antibody mediated, also known as humoral immunity. This is going to be the B cell column. Okay, so what we're going to see is a microbe. This could be some sort of virus, again, that's lingering around. Maybe it's a coronavirus. So these antibodies are going to come in and respond to the presence of these microbes. So we're going to try with the presence of our antigens. We're going to see the B lymphocyte coming in and secreting an antibody. So this antibody is going to detach from the antigen on the surface, and it's going to kind of float over and attach to one of these little spikes on the surface. And then we're going to see it in a little bit. There are multiple ways that these antibodies can affect or manipulate, destroy, get rid of, isolate, freeze, whatever it might be, certain microbes. So we have multiple different types of antibodies that affect and attack multiple different types of microbes. So we do see receptor site differences. So anyhow, these B cell antibodies are in the blood plasma. And again, they're there to block infection, eliminate infection. Okay? So that's antibody-mediated or humoral immunity. Cell-mediated immunity, this is going to be your T cells. So we're going to see a couple different pathways of T cell activity. We have the phagocytes that happen in that second line of defense. These phagocytes are going to be part of that innate immune system. So basically what we have is maybe a lymphocyte that went in and started gobbling up some sort of microbes or pathogens, kind of holding on to them, waiting for a helper T cell to come in and either inject a little bit of some sort of chemical that's going to destroy. Again, could be like a kind of a poison to destroy the microbe. Basically, it's going to, it's kind of like a digestive enzyme. It's almost like, sorry about that. It's almost like the macrophage consumed the microbe or the pathogen, but it needed help to digest it. So helper T cells come in and help with the digestion of the microbe that's being done by the macrophage. So it activates macrophages, in essence, providing the macrophage with some enzymes. The other thing we can do is pull in some viruses. We may see a virus that has entered into the cytoplasm of a cell, which will trigger cytotoxic T cells to come in and basically poison uh, the cell. So this is a self cell that has been infected by a virus, and the cytotoxic cells come in and destroy our self cells. So they kill infected cells, self cells. So if we have a cytotoxic T lymphocyte issue of recognition, that could be lead to or be the definition of specific autoimmune diseases. So these guys can be triggered or activated when we don't need them. So, but anyhow, so that's the kind of the basics of adaptive immunity. Antibody mediated, antibody with a B cells. Cell mediated immunity, we're dealing with T cells. Antibodies are what we're going to take a peek at as we go. So anyway, lymphocytes, surface markers. Lymphocytes have surface markers. So again, surface markers, antigens, chemical signals. So lymphocytes are a type of white blood cell, right? Type of leukocyte. So they're going to be found in the bloodstream. Okay, how do we activate a lymphocyte? Well, again, these lymphocytes or white blood cells are going to have a little antigen receptor site. Okay, so remember, an antigen is a surface marker. So some sort of a microbe comes in, and the antigen receptor is going to read the code as basically, does the key fit into the lock? And if it does, that could trigger some sort of a reaction, whether it's the release of an antibody, so we can bind a couple of these and clump them. We have a secondary signaling mechanism that we see as well. So again, it could be from an infected cell, an injured cell. It could be from a microbe itself. So we have basically two ways of activating a lymphocyte. One is through this antigen receptor. Okay, so we've got, remember, an antigen is a surface membrane surface marker. Okay, and then we have another type of receptor that can pick up cell debris, whether it's cell debris from a microbe or cell debris from our own cells. That could also trigger a lymphocyte. So remember, lymphocytes, not only are they helping with destroying, recognizing and destroying pathogens, they also, again, help with vacuuming and kind of keeping things clean. So anyway, we could get some, we'll see this later, depending on the degree of antigen and type of antigen that's picked up. In other words, what kind of a microbe are we dealing with? How many microbes are we dealing with? That could trigger then a cloning effect to occur. In other words, we're going to release a lot more active immune cells. So anyway, B cells, I'm going to zoom through some of this. You can read through these slides on your own, but I'm not going to spend too much time on B cell development. We know B cells are born in the bone marrow. They mature in the bone marrow. They're released from the bone marrow. They can hang out in the spleen. They can hang out in even the thymus. They can hang out in lymph nodes. But B cells, and remember, they produce antibodies. So we're going to look at antibody molecules. We're going to see all antibodies are going to have 2 heavy and 2 light polypeptide. So remember, poly means many, peptide deals with amino acids or proteins. So this is a long chain amino acid, a long chain protein. It's going to have 2 light and 2 heavy. The light chains have what we call a constant region. Heavy chains have 3 constant regions, and a variable region. That variable region can loop and bind to maybe a carbohydrate, other antigens. Okay, so all antibodies are going to have 2 antigen binding sites and 2 complement binding sites. So we'll look at that. We see the antigen binding sites on the end, and then we can see a carbohydrate chain that attached to one of these heavy chains. Okay, we see the heavy chains are going to be colored in all of this blue color. And notice the heavy chains are where we're going to see more of the binding going on. Heavy chains, thicker, more meat in essence, more chains or links to be able to attach items to, whether it's other antigens. So we look at an antibody, and we can see, again, it's got kind of your basic antibodies, and have Y shape. We've got some sulfide connections going on here, sulfur bonds. You can see the heavy chains. Again, these are going to be the complement binding sites and the antigen binding sites. Extreme diversity within our antibodies. We classify them now as immunoglobulins. So we abbreviate that with IG. So IgMs are going to synthesize and insert into their own plasma membranes. I had something written down here. IgMs. Oh, just basically associate IgMs as being naive. They connect with their own antibodies or their own plasma membranes. So antigen connection with themselves. So they can fold onto themselves. They can connect. I'll show you. It's AGM. You can see they've got multiple spikes or wheels. So, anyhow, let's see, IgGs, these are the bulkier of the group. About 75% of the antibodies in the blood are going to be IgG. And again, antibodies are secreted or released. Chemicals, they're proteins that are released by cells, and they can be released by antigens that are on surface. The idea is that the antibodies are going to be binding to antigens. Okay? Remember, antigens are surface markers on cells. So these are going to have specific shapes that are going to be able to plug in to antigens. So antibodies are chemicals, they're proteins. We call them immunoglobulins. Globulin meaning they're globular proteins from the immune system, and they're floating around in our bloodstream. They're produced by B cell lymphocytes. So lymphocytes release antibodies, specifically B cell lymphocytes. Okay, so B cell lymphocytes release antibodies, and antibodies come in different letters: A's, A, D, E, G, M's. These are immunoglobulins. The bulk of immunoglobulins that are produced and released by B cell lymphocytes are IgG. Okay, don't know if I had any other. Heavy, 2,2 heavy light polypeptide. Oh, increase. No, so yeah, we're good with that. High numbers, IgAs. Um, uh, you're going to find these mucous membranes, tears, saliva, IgAs. I don't think there was anything else I wanted to mention about those. IgE, these are your allergies, a small amount produces harmful effects. If they're too much, the IgE levels are high. That's a response to it's an allergic reaction or an inflammatory response. So anyhow, IgE, allergies. IgD, smallest amount, precise function is unknown. So there's a few of the different types. See the heavy chains make up the primary backbones, and then the light chains. Functions of antibodies, again, we get the antigen antibody reaction. We're going to see a couple different methods that this occurs, but basically their antibodies are trying to distinguish self versus non-self antigens. Okay, so in other words, self versus non-self cells. Okay, so the way that these antibodies can identify one cell from another is by the surface markers. What sign is hanging up on the door of the cell? And these antibodies are able to, in essence, recognize and fit into what these specific antigens are. And if they fit onto one and it's not recognizable as being from you, then it's a foreign or non-self cell. And that's gonna, the idea is that that they've probably already been triggered. So a lot of these antigens or these antibodies are going after different pathogens because they've been triggered by the first line of defense, second line of defense being breached or broken. Because these, remember, are third line of defense. These guys take a little while to show up. They can take anywhere from 3 to 5 days before they're actively attacking and destroying, triggering even a further immune response, which I'm going to get into next, is this image here, this illustration, I should say here. So we have an antibody here, and then this isn't an antigen per se. What it's showing is that these two triangles are the antigens. This red disc is going to be the cell or the cell surface, the cell membrane. So anyhow, these antibodies have little receptor sites, their little keys and locks. So basically, we're going to plug in the key. Does it work? If it does, depending on what type of antigen we have and what type of antibody we have, it could inactivate the antigen. So it could do something as simple as pull this grabs onto this side grabs onto this, and we pull it together and inactivate it. And then at that point, some of these are steps that lead to another. So I can inactivate it, and then I could bring it down here and get a phagocyte to consume it. Maybe I only want to inactivate it for a brief period of time. So this could be a chemical or a cell that produces or releases chemicals that we need, but we don't need them at the moment. So the antibody may inactivate the cell or inactivate the antigen, thus inactivating the cell for a brief period of time or even an extended period of time. So it doesn't necessarily want to destroy it. It just wants to inactivate it. The other thing that we see quite a bit, especially when we do blood typing, is this clumping, the surface antigens and then the anti-A antibodies are found in B blood, and the anti-B antibodies found in A blood. And if we mix an A blood with a B blood, we could have the anti-A antibodies coming in and binding to the surface markers that are located on the red blood cells, and that causes clumping or agglutination. So we'll look at this, and I think you should have done a little bit of blood typing in general, or at least discussed it briefly. You have a PhysioX that goes through it as well. So anyway, these antibodies can also pull in an antigen, bring it in for phagocytosis. We can attach an antibody to a mast cell of some sort. It's kind of like a two-step process. Maybe this mast cell could be a cell that has a lot of peroxisomes or lysosomes that need to be released, some sort of a chemical that needs to be released. That'd be like an immune system cell of some sort. And in order to activate this cell, we need to plug in an antibody and then bring in, or as soon as an antigen recognition occurs, soon as antigen recognition and connection occurs, it's going to initiate the release of inflammatory chemicals. So anyway, that second line of defense and third line of defense, it's not an on-off switch. There's crossover going on, and this is an example of that. Same with the phagocytes. We see the mast cells and phagocytes part of that second line of defense, and these antibodies part of the third line. Well, they can come in and attach and basically piggyback and open the doorway for these cells to release their inflammatory chemicals. Now, the last one up here is the one that we're going to talk about a little more coming up, is complement cascade. We mentioned that complement or that cascade effect. Basically, we're going to get a rush of a variety of chemicals. This is what's going on or can happen with COVID-19 after that 6 days of just kind of lingering, and our body is remember, epithelial cells can reproduce and regenerate at a fast rate, within a day or two. So over 6 days of it lingering in the system, the epithelial cells that it targets are constantly being regenerated. And so the body's, after a couple days of infection, the body's starting to send in some antibodies. But unfortunately, because it's still lingering and there's so much of it lingering around, that's why too the more exposed one is. So folks who are in nursing homes and are around it a lot, or nurses or doctors working in the medical field that are around it a lot. If you're in an enclosed space, for multiple hours at a time, like we're seeing people hanging out at the bars or at wedding receptions or public places, and they're there 3, 4 hours, 5 hours straight, and they're breathing it in and breathing it in and breathing it in, and the amount of exposure, and then it's lingering for 6 or 7 days, maybe even up to 15 days. Some people are seeing effects even quicker after like a couple of days. So that's why the contact tracing is very difficult for this, and figuring out what the timeline is. But so anyhow, they do know it's slow reacting, and it what that does is, like I say, it enables that innate immunity of epithelial linings to just keep regenerating and regenerating. In other words, providing basically an endless supply of target cells. So what that does when we normally, even with any type of infection, we're going to trigger a complement cascade as part of that second line of defense. And that's going to lead to inflammatory process, increasing heat, increasing blood supply, increasing the amount of possibly the amount of antibodies that we can bring to the mix. But in the meantime, because remember, they take 3 to 5 to 7 days to show up typically. So in the meantime, we're bringing in more chemicals to promote inflammation. Um, they call them cytokines as well. And we saw these earlier in the first video on immunity with inflammation. But anyway, there's so many different layers and types of chemicals involved with that inflammatory process. So what we end up seeing is, again, with COVID, that first line and then that second line, that 3 days, 6 day, 9 day period, is triggering and it's kind of stuck in that loop of the first to second line. So we get an extreme amount of what they're calling a cytokine storm. So we're getting that extreme inflammation, and eventually the amount of mucus and the amount of cellular membrane destruction and regeneration, regeneration. It's just perpetuating the cycle of target cells for the COVID-19 to continue to affect. So anyhow, we're trying to figure out how to stop or slow the adaptive immune third line of defense from kicking in, so we can basically allow that first line and of epithelial regeneration to go and then die out and not have the target cell availability, and again, waiting at 6-7 days, somehow delaying the third line of defense to come in. They think might be an answer. The other thing too, and we'll look at this further a little bit later in a different unit, is the ACE2 inhibitor trying to somehow not allow the connection of the coronavirus spike to basically not. So we want to keep the epithelial barriers, but we want to decrease or minimize the amount of receptor sites that it attaches to. There's a third player, and those are ACE2 or ACE2 enzymes, receptors. Okay. Angiotensin converting enzyme 2. And what that does is it lowers your blood pressure. It keeps you healthy. ACE2 is extremely important regardless of coronavirus. And so we find it all over the body. The ACE2 are on cells everywhere in the body, particularly though in the lungs and in the small intestines. So if we can somehow block COVID from attaching and activating these ACE2 enzymes, that could slow its progression as well. There's a third player that it's got a long, it's like a disulfide. We'll talk about it in more detail, but in essence, it's kind of like it's like a primer, kind of buffs the spike. They call it, the spikes on COVID are called S proteins, spike proteins. So they're the markers, they're the antigens. So these S proteins or spike proteins are the antigens on the coronavirus. We have a specific chemical enzyme in the body that actually will, that for how we don't know how COVID came about, but somehow one of our own enzymes will come in and buff and kind of clean off the S protein and basically prime it to attach to our ACE2 enzymes on our cell membranes. That perpetuates the process of the infection. So if we can prevent the crowns or the spikes from attaching to the ACE2s, then we can slow or stop COVID. We're having a hard time figuring out how to do that. So they've found and just recently discovered that there's another chemical that actually buffs that spike and then primes it, so our ACE2 will accept it even more readily. So if we can eliminate or get an off switch for that enzyme, then maybe we can get somewhere. So anyway, I don't want to belabor it. I've probably said too much already about this, because we really don't know much of anything. As much as we know, there's plenty more we don't. So anyhow, that complement, again, 20 different protein compounds. These are complements. So what they do is they are able to assist the immune system, the secondary line of defense, into ultimately the third line. But one example is this membrane attack complex that will go in and basically bore a hole into a microbe membrane, and what that'll do is allow for sodium, for instance, to rush in. And then remember, water always follows sodium, so that will rush in, and that could cause lysing or rupturing of the cell. We also see with a bacterial cell, when the complement is triggered, again, this is second line of defense. Complement is innate. We're born with many of these enzymes and chemicals, and we're also born with the triggering ability of the complement. So now, can some of these chemicals become more specific to specific bacteria as we get exposed to more pathogens in our lives? Absolutely. So in that way, there's some adaptive aspects as well. So anyway, we see the complement coming in, and we've got a bacterial cell, and these complements are going to form that membrane attack complex. Okay? So basically, they're going to work together, form a pore, and that pore is going to allow for more water to rush in, therefore more sodium to rush in, and thusly leading to some sort of lysing or explosion event. So C3, one specific complement, so this would be part of your innate immunity pathway, where, again, these don't need any type of antigen antibody stimulation. They can bind to bacteria or viruses without being triggered necessarily. So this is the second line of defense of that alternate pathway or the innate pathway that we see when it comes to bacterial or viral infection. These these come from mom. These are, again, part of the innate immunity. They also come from breastfeeding, we're finding as well. But anyway, these also have, we'll get back to that, or we'll talk about that later. I was thinking about the ACE2, angiotensin converting enzyme 2, has a zinc molecule attached to it. So anyhow, ACE2s are extremely important day to day for keeping our blood pressure normal. They're basically the parasympathetic aspect of lowering our blood pressure after a sympathetic response. So anyway, my brain is on COVID, of course, as all of yours are. It's on the immune system. It's on the renin-angiotensin aldosterone system, and that's coming up later. So if it seems like I'm getting ahead of myself a little bit, I think I kind of am. So, but I do want to introduce you to some of these terms because and concepts, because we'll see them again. Some of the ones we're talking about now, we're going to see again. So I've said it before, said it throughout. We are putting a huge puzzle together, and a lot of these ideas and concepts start to make more and more sense as we go. And we're about halfway through, so hopefully some of them halfway through 6 weeks of A&P have sunk in. So anyway, primary response and secondary response. Again, that primary response, it comes after a stimulus, and it can take up to 10 days to get these antibodies released. And notice, they're relatively, they'll maybe hang out for 3 to 5 days, and then they start to dissipate. Okay, IgM and IgG, that's from an initial stimulus. Now the same microbe, we get secondary exposure. So we've been exposed again. Notice how quick, maybe only 3 days to respond as opposed to 10. And then we've got about the same amount of time, maybe 3 days of release. Notice with IgG, after day 12, we finally get it to respond. Well, now it's like after day 4 or 5, we get it to respond. And then we get a long sustained release of IgG. So anyway. What else was I going to talk about? Oh, clonal selection theory. I think I had in my notes just to mention that, again, we have a diverse set of specific antibodies available. And we have the precursor, in essence, the stem cell. As these mature, we're going to start seeing clones created from the mature lymphocytes. So we have a specific type of B cell here, B cell here, B cell here. So we have 3 different types of B cells that will produce 3 different types of antibodies. So we can see we've got different shapes of surface markers on these antigens to be able to identify other antigens. So if we get one of these to link up with a microbe that's going to trigger some sort of immune response, we're going to get this cloning, antigen-specific cloning. So that's, and again, this takes 5 days or so to get to this point. So when we have an infection, day 1, day 2, target cells are really getting affected. We're kind of waiting on the troops to arrive, and then they do, and they destroy what microbes are lingering. And then the epithelium, or that first line of defense, the mechanical and chemical barriers, they start to remodel and wake back up. Okay. So new target cells are being made. But by then, typically after 5 to 7 days, they're starting to rebuild. By then, the virus or the bacteria has been eliminated typically. And again, with COVID-19, we're not seeing that. We're seeing a different, more prolonged state. So after a week of being infected, your body is still producing, just like it normally would, more target cells. But the virus is still there. Usually it's eliminated by day 3 to 5. It's still lingering at day 6, and we're still providing it with target cells. So anyway, so that's clonal stuff. T cells, again, mature in the thymus gland. T cells become either memory T cells or effector T cells. The memory T cells can have, basically they will change into being an effector T cell. So these guys basically are waiting around. They have a memory of what kind of pathogen they're able to destroy. And once the body triggers the memory T cells to come out of hibernation, they convert to effector T cells, and that's going to cause, they're going to plug into the virus or the bacteria. That's going to trigger cytokine activity. We have cytotoxic, and again, cytokine activity is the inflammation mediators. They make the area inhospitable. So this is third line of defense, again, working with the second line of defense. Second line, we got some inflammation triggered. Third line comes in, and at times we want to perpetuate or continue that. We call these pyrogens. We want to continue that, generating fire, generating fever or inflammation, and certain T cells are able to do that. Some T cells, again, they're going to plug in and inject a poison into an infected cell and destroy it. Helper T cells are going to be called on when B cells need help, phagocytes need help. So just like their name sounds, helper T cells, they regulate the function of multiple different immune system responses. They come in to, again, kind of help out. Suppressor T cells, again, these are going to, if we've turned flip the switch to on, we have to, or crank the dimmer to bright, we need to be able to bring it back to dim. So suppressor T cells suppress lymphocyte function. This is what we need to happen potentially with again, certain diseases or disorders that are stretching out, such as the coronavirus. HIV is another one that just kind of lingers, and it's your body cells can never really tolerate what's going on. So suppressor T cells they help with promoting what we call self-tolerant, immune balance and regulation. Because again, if you have triggered because of a viral infection, you have this trigger of a conundrum going on where your immune cells just keep coming and keep coming and keep coming and keep coming, and we need them to stop. Sometimes we need them not to show up when they are, because we need these stages of I'll come back. We need these stages of adaptive immune response to be appropriate to whatever the virus is that we're infected with. And some of these SARS-related viruses, like COVID-19, so HIV is another one. We're having a difficult time delaying some of these stages or altering some of these stages. So anyway. So we see, again, at day 0 through 7 down here. So we get an infection. The first 3 to 5 days, these guys are just kind of getting prepared. We're awakening some of the sleeping T cells. We're waiting for these B cells to basically get prepared to show up and get the right kind of antigen antibody connection figured out. And so this takes a little bit of time. And so by day 3 or so after infection, we start to see cloning going on. We start to see more troops showing up, or not showing up maybe necessarily, but getting ready to show up. So the phone tree has essentially kicked in, that we've got a problem, and so we need to start cloning. And then we're going to get some differentiation. We're going to see these effector Ts. Basic differentiation means these guys are going to start separating away from each other and starting to show up onto the scene. So by we call this the activation phase. So by day 7, we've got an adaptive third line of defense showing up. Okay, and then for about a week or so, we're going to see humoral immunity. So these antibodies going after antigens, this is the B cell lymphocyte activity. And then the T cells are going to continue to plug in and either spritz what they need to do, the cytotoxic T cells, and that causes, again, that apoptosis. So that's the decline of being infected. And so we're going back to homeostasis. And now we've got the surviving members of the Bs and Ts. And so those are going to be the memory cells. Okay, so they're going to go back and they could potentially trigger some of their neighbors to show up. We do see that going on as well. Types of adaptive, natural immunity, non-deliberate exposure to antigens. So this is being basically you're in the womb. Okay. Or you get coughed on once you're out of the womb, so environmental. And then artificial is deliberate exposure, maybe through immunization. So we do see natural, passive, for instance, mom to fetus, mom to baby, and breastfeeding. Active, artificial. So the opposite of natural is artificial. The opposite of passive is active. Vaccines and immunizations. You can click on this and it'll take you to a nice little website that breaks down some of this information as well. So anyway, we're just about done. We've got immunity is broken into adaptive and innate. Okay? Innate immunity, primarily thinking some of these first lines of defense, these mechanical and chemical barriers. We're thinking of phagocytes and phagocytosis. We're thinking of cascade elements. We're thinking of NK or natural killer cells. With adaptive immunity, we're thinking more like the B cell, T cell, lymphocytes. And from adaptive, we either have natural or artificial. And from there, passive or active. So maternal is natural, so from mom. And again, through birth and breastfeeding and being in the womb. And then active, again, an infection of some sort, somebody coughs on you. Okay. So this is what we're seeing. COVID is this active, natural immune system that's getting triggered. Okay. Artificial adaptive immunity. We have passive artificial. This is rare. This is someone gets infected with something, and then we transfer antibodies after the fact. Primarily, we think of active artificial immunity being active through the means of immunizing. Okay. Stages. This is just what we went through. So anyway, I'm going to close and leave it at that. I think we've talked and done more than enough. Again, I want to reiterate that Unit 6, Module 2 is coming to a close. Module 3 and Module 4 will be open. And so anyway, we're getting we're halfway through. Please complete that midterm survey. Those of you who have, thank you. I appreciate it. And again, email me with any questions. If any of this is confusing, email me and let me know. You also want to go in and look at the. Let me do a couple of things real quick here. Go back into our Blackboard. A couple of things that I just want to clear up. we had some glitches, as you know, with the videos I mentioned earlier. These are now thumbnails that'll take you directly to YouTube as opposed to Panopto. And again, the other thing you want to look at when you go into units, module 2, okay, and then underneath your practice area, you're going to find immunity review questions along with the Quizlet. So go through your Quizlets. Immunity review questions will again open up a document that'll have several of the types of questions that you're going to see on your exam. So that's what's in that document. Again, I don't think it shows you the choices. I don't know if it can, or I don't think so, but it does give you the questions that you can see what the questions look like at least. Yeah. So yeah, it doesn't show you which of the following is true of lymphokines. So what I would do is just look up what is a lymphokine. I mentioned it briefly in the first video. Which substance can destroy pathogens by chemically hydrolyzing them? Okay. Which substance can lower pathogens by lowering the pH level? Okay. These are all in the complement system activated by what? Okay. So go back through the PowerPoints, review the notes. And by the way, too, when I say view the notes, if you click on if you click on a PowerPoint, so you go in here, you want to look at the one that we're doing right now, it's going to open up something, I believe, that looks like this. There are no notes there. So you need to go up here and click Open with Google Slides. And then from there, you'll be able to see the notes. Okay. Another way you can see the notes, if you click Present, and then you click Notes, it'll pop it up this way as well. Oops. So you can split screen it, or you can view the notes that way. You can just click on the different. Okay. So that's another way that you can view the notes. Okay, but again, if you just click on it, it'll take you to the PowerPoint like this. So you do want to open it in Google Slides. Other than that, again, how do you get to the survey? Just go to announcements, click midterm course survey, and it'll pop it open. And you go through and take the survey. Anyhow, that's it. Have a great 4th of July. And if you have questions, please email me and let me know. I look forward to getting to implementing some of your suggestions regarding moving forward with exam prep and that, and getting these videos out for you. Okay, so I guess that's it. Figure out stop recording. Okay, have a good day, everybody.